much for joining us. I am thrilled to kick off our series this fall with two excellent presentations by my colleagues. Hong Ji, assistant professor in the School of Veterinary Medicine, and an expert in the biology of epigenetics in pediatrics, and Professor Janine Lassalle from the School of Medicine, who is also in the Department of Microbiology and Immunology, and also an expert on epigenetics. And both of them will talk about implications of DNA changes in response to environmental exposures. I believe before we start the conference, Ana Lucia has a slide for me to make an important grant announcement. These calls just came up, and we thought that it would be really interesting for a number of us here in the audience. One of them is an innovation award on COVID-19 and health equity. It's a UO1 call, and it can include clinical trials. And I think Ana Lucia mentioned that there might be some collaborators who would like to get connected with some of you and work on this grant.
Correct. So I just want to highlight that for this particular opportunity, the Center for Healthcare Policy and Research is interested in submitting a proposal, but they are looking for collaborators. So if you would like to connect with them, you can email Belinda Martineau directly. I will post her info in the chat, or you can send me an email and I'll connect you. If you are interested in this opportunity or any other opportunity and you are looking for collaborators, please let us know how we can help you connect with people inside and external to UC Davis.
Thank you, Ana Lucia. That's wonderful. And then the other call is also very relevant. It's a pediatric COVID-19 data challenge. And it's from the Department of Health and Human Services, and it's a BARDA application. So please reach out to us and especially to Ana Lucia if you need additional information and check these grants out. And without any further delay, I would like Hong and Janine to start the epigenetics session.
Can you hear me?
Yes, very well.
Thank you, Angela, and the other organizers of this forum for the opportunity to speak here and share with you some interesting data our group recently got on suggesting the long-term epigenetic impact of wildfire smoke exposure. My name is Hong Ji. I'm currently assistant professor from the Department of Anatomy, Physiology, Cell Biology. I'm also part of the California National Primate Research Center. So I study epigenetics. What is epigenetics? So epigenetics means, on top of genetics, it refers to modifications of DNA or associated factors without changing the DNA sequence itself. This information or these modifications are maintained during cell division, and they can cause stable changes in gene expression. So it's often viewed as a mechanism of regulating gene function. I like the image here down here captures a few important epigenetic modifications within the nucleus. So the brown ball here shows the DNA methylation, which is the addition of a methyl group to a cytosine. And there's also histone post-translational histone modifications. Here in the blue ball here is the active, is a histone modification associated with active transcribed genes. And there's also repressive histone marks associated with the histone chromatin, heterochromatin. You can see here the chromatin, I mean, the nucleus is also organized into this, the regions with more accessibility and the regions with less accessibility here. And this nice structure in normal cells is lost in disease, such as in cancer cells. For example, that you can see that this nice compaction of chromatin organization is lost in cancer. And this could be due to mutations in epigenetic modification enzymes, such as the DNA methyltransferases and all that. Also, it could be due to activation of epigenetic modulators. And all of this could lead to stochastic gene expression. And in the case of cancer, it could promote the cancer cell proliferation and survival. And numerous studies have linked exposures to environmental factors to changes in the tissue and cell-specific differences in the epigenome. And it's also important to note that the pre-existing epigenetic landscape could also affect the response of a cell to environmental exposures. So this interaction between the epigenome and the environment could be two ways. And this is one. This is why people are interested in studying the epigenetics, because it could be a mechanism to mediate the gene-environment interaction. And my group, one of the focus is to a research group is interested in studying the health impact of exposure to particulate matter and the role of epigenetics. And the particulate matters, especially those with smaller diameters, like for example, PM2.5, with the particles that's smaller than 2.5 micrometer in diameter, could get into the alveolus of the lungs and enter the circulation and pass on to other organs. So the particulate matter or smaller size of particulate matter have a multi-organ effects, including it could increase risk of pulmonary diseases and cardiovascular and neurological disease. Besides generating systemic inflammation, it could also have some gestational outcomes, such as smaller birth. And these particles could come from many resources, including traffic, in this case, a diesel engine, and also wildfire. So it's important to know that early life exposure to air pollution, PM2.5, has a long-lasting impact on respiratory health. And this is because lung development starts in utero, but it also continues significantly, continues after birth, including significant alveolar development and lung volume increase. And it has been shown that air pollution during this period can increase, reduce lung function and increase risk for wheezing and asthma. And these two are significant predictors for fixed lung air construction and COPD in early adulthood. It's also important to note that early life is also a critical window for immune programming, both because of the dysbiosis, the colonization during early life, as well as the interaction between exposures, the lungs and the immune cells. So this really speaks to a developmental origin of lung disease, which is part of the DoHaD hypothesis, that the environment and individual experiences during early development can affect their sensitivity or risk of developing disease later. Here, again, as I mentioned before, lung development starts about 4 weeks in utero and continues after birth till young adulthood. So you can imagine that any exposures that could change the epigenome during this phase could potentially dysregulate genes and lead to increase the chance of developing diseases in the lungs. In addition to this development, dynamic development for the lungs or the organs, the epigenome itself also undergoes dynamic changes. So this figure, the y-axis shows the levels of DNA methylation, while the x-axis marks all the different developmental timelines from fertilization to birth and then to adulthood. So you can see here the methylation really undergoes dynamic decrease and increase waves of changes before birth, and even after birth continues after birth. And it's also shown that during aging, the epigenetics changes too. And there is this epigenetic clock defined by DNA methylation that is different, often different from the chronological age because of disease state or exposures. So it has been shown, many studies, including this one, very nice one, demonstrated that early life exposure to PM2.5 really increases the risk of asthma, and the epigenome plays an essential role here. So in this study, the authors exposed mothers of mice. This is in mice. This is a mouse experiment. So they exposed F0, which is to environmental particles, and then established asthma in F1, F2, and F3 by exposing them to allergens. And they showed that maternal exposure really increases risk of asthma development in all 3 generations. And interestingly, they also find epigenetic changes in splenic dendritic cells in all 3 generations. Importantly, when they treat the F1 mice with an inhibitor of DNA methylation, which inhibits DNA methylation genome-wide, the asthma phenotype in F2 and F3 were abrupted. So this really supports that epigenome, in this case, DNA methylation, plays a mediating role in connecting the early life exposure to PM2.5 to the offspring, the development of asthma in offsprings. However, the studies of postnatal early life exposure to PM2.5 and epigenome are lacking, and we, as our research group, have shown that early life exposure to traffic-related particles was associated with changes in DNA methylation in children. And here it shows the correlation, one particular CpG site, DNA methylation site, that the methylation levels is associated with levels, is positively associated with levels of exposures to traffic-related air pollution. And the open circle is are non-asthmatic children, while the closed triangles are asthmatic children. And interestingly, the same methylation, the methylation at the same site, also positively associated with symptoms, the frequency of symptoms, which is one way to measure the severity of asthma in children. So in California, especially, then another one big significant contributors to PM2.5 is wildfire. And this map shows the map of wildfire smoke plume extent of last year when the lightning complex fire started. And this is the picture of fires and smoke plume extent as of last month, end of last month. You can see here that not only the fires accumulate, the smoke accumulates at the site of fire, but it also travels to in long distance, which means that a lot more people are affected by wildfires as we originally thought. And then in California, it is estimated that about 50% of PM2.5 is from wildfire smoke. And emerging studies have demonstrated that short-term effects of wildfire smoke exposures, including respiratory symptoms across all ages. However, the long-term effect of wildfire smoke are just start to be understood. Our collaborator, Dr. Lisa Muller, have utilized the study in rhesus macaques and find that wildfire smoke exposed monkeys had reduced lung function compared to control animals. And these animals this wildfire smoke exposure actually happens when they were babies. And then the reduced lung function happens when they were adolescent and young adults. And then her group also found that the blood cells from wildfire smoke exposed female animals had reduced IL-8 protein levels when they were treated with bacteria components, ligands, while the male animals, the wildfire smoke exposed male animals, had reduced IL-6 protein levels compared to controls. And both IL-6 and IL-8 are important pro-inflammatory cytokines in lung diseases. However, at baseline, they did among the 84 toll receptor pathway genes they've studied, only 2 showed some moderate differential expression when there were no treatments. Lastly, there was some unpublished data from her group showed that this effect of wildfire could be could pass on to the next generation, and the offsprings of wildfire smoke exposed animals showed different immune profiles compared to the offsprings of control animals. So based off all this data and the proposed role of epigenome in mediating the impact of early life exposures, our overarching hypothesis is that exposure to wildfire smoke during early life causes persistent epigenetic changes in the airways, which alters immune and lung function and increases risk of asthma and other lung diseases. And this is to utilize the same cohort that Dr. Miller has been following for years, and which includes a 2008 cohort that were exposed to wildfire smoke when they were less than 3 months old. And then there's also a 2009 control cohort, which are born around the same season, but without wildfire smoke exposure when they were young. And their group, as I just mentioned before, they studied peripheral immune inflammatory responses, lung function in both adolescent animals and the young adult animals, while we collected nasal samples and studied their epigenome and transcriptome using whole genome bisulfite sequencing and RNA-seq. And this is work done by Lucy Tsai and Anthony Brown. So here shows really captures the average daily PM 2.5 over time in the animals we studied. And this is the peak that come from the Trinity County fire that happened in the summer of 2008, early summer of 2008. And if you zoom out, this shows that during this peak, it really contains like about 10 days of high wildfire smoke exposures in these animals. And so we did a whole genome bisulfite sequencing, and we found that we identified over 3,000 differentially methylated regions that are different between exposed animals, which are showing red here, and then the control animals. And the majority of these differences are more methylated in exposed animals compared to the controls. And these DMRs are annotated about to 2,137 genes. And we further looked at where these wildfire smoke associated DMRs are located, and we found that they were significantly enriched in promoters, exons, and depleted in the intragenic regions. And in terms of CPG, they are also located at CPG islands, shores, and shelves, and depleted at open seas. And because the DNA methylation often regulates the binding of transcription factor, we also looked at the enrichment of binding sites among these DMRs. And we identified 181 enriched TF binding sites. And top 10 are shown here. Just to make, and then, among this top 10, 6 of them belongs to the 8 of them belongs to the BZIF TF family, which are has been shown that DNA methylation inhibits their binding in vitro. And consistently, we also find that among the ChIP-seq data sets for this transcription factor, a lot of them are either unmethylated or partially methylated, which means that this means to us that potentially the more methylation induced by wildfire smoke exposure could potentially inhibit the binding of this transcription factor to their targets. And then we continue to look at the chromatin stage around these regions with DMR, with wildfire smoke DMRs. And we found that there was 15 chromatin state we checked, and we find significant enrichment for markers associated with bivalent transcription start site and enhancers. And this indicate that these bivalent markers are often located at genes, key developmental genes that are often lowly expressed, but really poised for activation. The reason why they call bivalent is because they have both active marks and the repressive histone marks at the same time. And the two epithelial lines are highlighted here. And then we looked at significantly enriched pathways among the differentially methylated regions, among the genes associated with these DMRs. And we identified over 100 pathways, and top 10 are shown here. If you pay closer attention, then you can see that some of them are associated with brain function, like exon guidance signaling, synaptogenesis, and Krebs signaling neurons. And I also highlighted a few pathways that's as examples of immune responses or processes. We also looked at whether these wildfire smoke associated DMRs are also overlaps with potentially DNA methylation markers for lung diseases. Among these 5 studies, they've used a rate-based method, which covers much less than our whole genome bisulfite sequencing, but they studied DNA methylation marks associated with COPD, IPF, and asthma. And we're very excited to see that there's some overlap between the wildfire smoke DMRs and these DMRs associated with these lung diseases. And here I'm going to show you like 2 examples of DMRs, how DMR look like. In this case, it's a DMR associated with, located at IL4R, and it's more methylated, which is in red here, in exposed animals compared to the controls. And this gene is a receptor for IL4 and IL13, which are Th2 cytokines that are important for many aspects of airway inflammation and airway remodeling. Another example shown here is a DMR associated with, located at a Toll-5 receptor, the Toll receptor 5. And it's again more methylated compared to controls in exposed animals. And this receptor is a receptor for flagellin, and it's upstream, and the activation of this binding of this receptor could produce IL-6 and IL-8, which are pro-inflammatory cytokines I mentioned before. Our RNA-seq analysis only identified 1 significant differentially expressed genes between the exposed animals and the control animals, and this is FLT2, and this gene is downregulated in the exposed animals. And this gene is an interesting one because it has been shown that it can protect lung epithelial cells from oxidative stress-induced apoptosis. And then by integrating the epigenome or the methylome and the transcriptomic data, we correlated them and it found that methylation at 172 DMRs significantly correlate with gene expression. And here is one, although it didn't meet it's not differentially expressed. However, we found that for this hypomethylated, like with less methylation in exposed animals, there's a positive correlation between DNA methylation and gene expression. And this gene is part of the Wnt signaling that's known to participate in utero lung development and early life alveolar genesis. Lastly, we explored the data set to identify cluster. Instead of looking at individual genes, we looked at clustered of genes that could be associated with wildfire smoke exposure. And we identified a purple cluster here that is marginally significantly associated with positively associated with wildfire smoke exposure. And this cluster contains about almost 600 genes. And we find 21 significant enriched pathway in this purple module. And top 10 are shown here. You can see that several viral related gene antiviral response and immune response are highlighted here. So in summary, we identified a genome-wide differentially methylated region associated with early life wildfire smoke exposure in nasal cells from rhesus macaque. And the majority of them are more methylated in exposed animals. And these DMRs are enriched genes involved in immune response, brain function, lung development, and they're implicated in gene regulation of key developmental genes. And one possible mechanism is that it could affect the methyl-sensitive transcription factor binding. And we also identified one FLT2 that was significantly expressed between exposed and non-exposed animals. And we also observed significant correlation between 172 DMRs and expression of genes, neighboring genes, supporting that these methylation differences could impact gene regulation. Some ongoing efforts. We are collaborating with Dr. Rebecca Schmidt to conduct a pilot study to see whether we could see similar associations between wildfire smoke exposure and the epigenome in young children. We are also trying to establish cell culture animal models of wildfire smoke exposure using particles collected during wildfire, and this is in collaboration with Dr. Keith Ein. And our primary center also have a combustion facility in the inhalation core. We are trying to get that working. With that, I'd like to thank people in my lab, and this project really started right after the pandemic started and then completed well, and especially Lucy and Anthony significantly contribute to this project. I also want to thank our collaborators and primary center and on campus, especially Janice's group, to help us establish the analysis pipeline in my lab. Also the Environmental Health Center for their support of this project and to work with the community engagement core to and our community community partners to try to disseminate this information to the community. With that, that concludes my presentation. Thank you.
Thank you, Hang. I think we should postpone the questions for after Janine's talk. In the meanwhile, we could still chat in the chat feature. And Janine, you are muted.
Okay, that helps. Okay. Well, thank you for the invitation to talk today. Yes, I've been on the Davis campus for the past 24 years on the Davis side of the med school. And it's always the team science aspects that keep me happy here. And so today I'm going to be able to talk to you about a recent success story we've had with the team science approach, looking at DNA methylation in cell-free fetal DNA to get at potential, the impacts of maternal obesity on the developing brain. And again, this is my departmental affiliation, but in addition, a number of the research centers here have been instrumental to both this and our other research endeavors. So why should we care about epigenetics? I think Han gave a great introduction, but I just want to kind of point out that our environment is dynamic. You know, in addition to the global pandemic, every day in the news, we see evidence of our changing changes due to climate change, floods, fires, smoke, heat. And what I'd like to point out is epigenetics, understanding epigenetics may be really key to improving our ability to adapt to this changing environment, as well as to address important issues of environmental justice and the impact on future generations. And I know sometimes people like to put genetics and environment in two separate boxes into some convenient equation, but as epigeneticists, and you saw this from Hung's talk, that we really think about interactions between genes and environment. So I like to point out in this slide that there is like a two-way street as an analogy. On the one side of the street, oops, sorry about that. Fancy and funny here. Okay, you have genetic differences that influence environmental susceptibility. For instance, you might have underlying differences between all of us that affect how we respond to the same environmental challenge. You can have environmental factors that directly or indirectly damage DNA. But last but not least, you can have these environmental factors that influence gene expression through long-lived changes in gene expression at the level of the epigenome. So my lab is interested in bringing these genomic technologies to help solve this problem. And again, if you talk to an epigeneticist, there's no such thing as nature versus nurture. It's nature and nurture, or gene by environment interface. And again, it's very complex. If you think about complex human phenotypes or disease states, when you think about the genetics and environmental components that go into that state, you can have, again, multiple interactions. Some are one way, some are two way. You've got all this going on. It's almost impossible to get at all of those different factors. But what you can do with these technologies is take a cross section through them. And that's really what we're doing with epigenomics, genome-wide investigations, as well as things like metabolomics. You're also looking at that interface of all of the complex roads going back and forth. And by looking at that interface, you get insights into what's going on, and you can get insights into the G by E. And as well, this bringing them all together into multi-omics really helps you not only gain insight, but also potential develop biomarkers for risk of human diseases. And I'm not going to talk about this, but we're also starting to get into the AI machine learning aspect, because the goal here is using this insight or big knowledge, or big data to knowledge, to actually come up with predictive models or predictive biomarkers that could help gain insight into how to predict and potentially prevent some of these phenotypes that are involved in complex human disease. And so one of the big problems we're hoping to address with these technologies is that of obesity, and particularly maternal obesity, because maternal obesity not only affects, the mother, but also it increases the risk of childhood obesity and metabolic disease, and really the lifelong risk of cardiovascular disease in that child as they grow up to adulthood. And many factors contribute to the programming of offspring metabolic health, including placental nutrition delivery, epigenetics, and I'm particularly be talking about DNA methylation, but also inflammation and things like the gut microbiome. And overwhelmingly, this is a very common problem, because 2 out of 3 women in the US now are either overweight or obese. And it's also the point where probably the biggest health impact in terms of preventative healthcare in our country could be done, because preventative interventions to change diet or exercise in pregnant women could have long-lasting impacts, not only on their own health, but also their children. And as an autism researcher, I got interested in maternal pre-pregnancy, maternal obesity, because it was showing up in the epidemiology literature. And that is that maternal pre-pregnancy overweight, which is a BMI over 25, or obesity, which is a BMI over 30, increases risk for autism or developmental delay at about 1.5 to almost twofold times higher, which is a pretty big risk, although obviously not every child born to an overweight mother develops autism. So there's a lot of individual variation. Also, in addition, if you just look at the males, excessive gestational weight gain, so this is the weight gain during pregnancy, is associated with autism. And then this is striking because both obesity and autism prevalence have increased in recent decades. For autism now, we're at 1 in 54 children in the US are diagnosed with autism. And because there's a large male-female bias, about 1 in 36 boys versus 1 in 135 girls. And if you combine all of the neurodevelopmental disorders, so this would include intellectual disability and common things like ADHD, 1 in 11 children now in the US are diagnosed with a neurodevelopmental disorder. And so obesity prevalence probably doesn't need any introduction to this audience that obesity is increased in the US, but what you may not know is that this is actually a worldwide phenomenon now, and that actually women have a higher prevalence, particularly of severe obesity. And I just kind of throw in these graphs here because they're very striking. These are sort of lumped by different countries. The blues are the English-speaking high-income countries and European countries, which are known to have an increase in obesity, but also Latin Americas are in green. The Asia is yellow and orange, and then the African countries are in the reddish tones. And you can see a couple striking things. So here, this is obese versus severe obesity, and severe obesity is defined when your BMI gets over 35. You can see it's higher in women on the right than men, and it's worldwide. And so you might say, okay, well, this is due, the worldwide phenomenon is due to the influence of junk food and high fructose corn syrup, and certainly that must be a portion of the problem. But it's interesting to point out that animals, both in research settings and the wild, have increased in this. And this was a sort of very prescient publication that came out in 2010, saying titled as "Canaries in the Coal Mine," reporting on weight gain in animals, both in research settings, and you can see on here there's a variety of different. These are all mammals. And the way to read this graph is that everything that's up in this right-hand quadrant means that there's been an increase in the weight of these animals over decades. And you can see they're all in the upper right-hand quadrant, which means they are all increasing over recent decades. And chimpanzees are out here on the extreme, but you can see all animals are there. And although it's less striking to look at it, they did in this paper looked at the sex difference and found a significant difference that the females were more susceptible to this than males. But this is also an interesting sort of natural experiment for the one I'm going to be telling you about, because if you see the macaques and macaques from several different research colonies, including the one here in California, are about in this range here. So they naturally get obese. And then so for the experiment that I'll be talking about, we were able to take advantage of that because this is the indoor colony of monkeys. Hungstock was on the outdoor colony. These are the indoor colony, and there's quite a few that just get obese in the same way that humans do. So yeah, this is the project that I'd like to share with you today. It's a great example of team science because it involves 7 departments, 4 schools, and 4 centers. Cheryl Walker is actually the PI who wrote the grant, and it was funded by NICHD. And I'm going to be telling you about our portion of it, which was the epigenetic portion, and that was headed up by my very talented postdoc, Ben Laufer, who now has recently left my lab to start as a senior scientist at Genentech. So here's the experimental design. So we had 4 different groups. So again, the obese animals, so these are obese females that were divided into either untreated obese, calorie restriction, which means they just got fewer calories during pregnancy, or treatment with a drug called pravastatin, which is a statin drug that's there to reduce inflammation. All of the treatments and behavioral analyses were done through the Primate Center, and this was headed up by Kathy Vandervoort, and the behavior being done by Melissa Bauman, Mary Gollop, and John Capitano, and definitely help from Casey and Jasmine on these. So what we did then is so there was blood drawn prior to mating, before pregnancy, as well as at 4 different time points that covered all 3 trimesters with 2 time points in late trimester. So we had blood that we used to get cell-free fetal DNA from. And then they were also measured for metabolites by Carolyn Slupsky's lab, lipids by Amir Taha's lab, and then immune markers or cytokines from Judy Vanderwater's lab. After the monkeys were born, these were all male births because we wanted to keep males are a higher risk for a number of the neurodevelopmental disorders. So they measured social behavior. Really, there was a lot of behavior measured in this period. I'm just going to be talking about the social versus abstract behavior that was done about a month old. And then at 6 months old, the animals were sacrificed in order to look at the brain region. So there were 3 regions of the brain where we were able to look at, again, DNA methylation compared to metabolites and lipids. So what is cell-free fetal DNA? I want to do a little introduction. This is a remarkable new technology that allows non-invasive prenatal testing for genetic disorders. And this is basically because the cells from the fetal trophoblast are shed in normal stages of apoptosis, and they release then the DNA into the periphery, and this is actually probably encapsulated by these vesicles, but basically it can be found in the plasma fraction of maternal blood. And this is currently used clinically for prenatal testing of things like Down syndrome. It's actually a screen that if you don't have any evidence, then they won't go on to an amniocentesis. And so this is really kind of revolutionized the way that prenatal screening is done for the genetic disorders, but it's also, being potentially used for epigenome as well as transcriptome. And again, to remind you that this is coming from it's fetal in origin, and the tissue is basically the trophoblastic cells from the fetal placenta. This fraction increases during pregnancy and ends up to be about 12 to 41% of maternal cell-free DNA is actually cell-free fetal DNA. And so going into this experiment, we were excited about this because my lab for a number of years has been interested in the connection between the placenta and the developing brain. And we really had this hypothesis then that could cell-free fetal DNA derived from placenta may have an epigenetic signature that reflects or predicts infant brain and behavior. And this comes from our previous mapping study of the placental methylome that showed it had a very distinct profile of early embryonic development, including relevance for neurodevelopmental disorders. And then previously we've shown in a human cohort, and then we have one published study and one that's currently under review, where we take a high risk cohort for autism and look at placenta prior to diagnosis of autism. And we're able to distinguish newborns that are later diagnosed with autism versus typically developing by their DNA methylation profiles. And then more recently, we have another paper under revision where we're actually looking at a mouse model where we can get placenta and embryonic brain. And this is environmental exposure of PCBs that again show that there's a nice link between placenta and embryonic brain. So from this experiment on the monkeys, we've got a lot of data. I just want to kind of point out again that, and again, I'm glad that Hong covered this because we're a very similar approach. We're using whole genome bisulfite sequencing to sequence the whole genome for DNA methylation. And we end up with a large number of these DMRs or DNA methylated regions. And there were a lot of different comparisons. So I'm not going to show all of the data, but I want to just hit the highlights. And this is one of the highlights we have, is that when we look at the DMRs that were isolated from cell-free fetal DNA or brain in the study, they actually overlapped with those we previously described in humans, and there was at least definitely not a complete overlap here. This is the fold overlap, and this is the Q value, which is the corrected P value, and you can see that for placenta. And again, reminder that the monkeys that we had were male, so we had a stronger overlap with the male placenta in human. This also has to do with the fact that of the male bias, we have very few female placenta ASD cases, but definitely we saw that the male overlap was significant. And then when we move into brain, and this was brain that we previously published for differential methylated regions for idiopathic autism, as well as 3 syndromic forms of autism, including Rett syndrome, Duke 15q, and Down syndrome. And here we get some nice significant overlaps with the monkeys for both brain DMRs as well as the cell-free fetal DNA. But in addition, we were looking at, again, these gene pathways that come out when you put all of the genes that are associated with the differentially methylated regions. And there was some striking overlap that we saw at the pathway level between what was isolated from cell-free fetal DNA versus brain. And again, this is coming from the placenta, but we got glutamate pathways, include a couple of glutamate pathways. Angiogenesis is really interesting. It came out in both the cell-free fetal DNA and brain, and that also kind of links with these EGF and PDGF pathways. Angiogenesis, the placenta needs that in order to go out. It's kind of like a cancer. It goes out and seeks blood vessels, and actually epigenetically, the placenta is quite similar to cancer in that way. And that also came out in brain. Brain is also important for the blood vessel formation. So the pathways that were overlapping were glutamatergic synapses, angiogenesis, and integrin signaling, and integrin is involved in cell adhesion. But I also wanted to point out a couple of really striking findings we have when you get down to specific gene loci. And this is a region that came up. So normally we're looking at fairly small regions of a few hundred kb. This is a 14 kb block on a whole chromosomal region of chromosome 20 that came out. It was very striking. There are a number of genes in here, but over this 14 kb block, let me just take you through these plots. On the y-axis of each one of these is the percent methylation. And then every spot is an individual nucleotide or CpG that's been, this is the percent methylation of that. And then the animals are represented here as color-coded. Red for the ones that were obese during pregnancy, blue are the control, and then yellow are the combination of those two interventions. And hopefully what you can really see is over this entire block, not only do we see a hypermethylation of this region associated with obesity, but the interventions are in that intermediate region. And for all 4 time points that cover all 3 trimesters, you see the same pattern. So this is reproducible throughout the pregnancy. And then when we look at brain from the infants from these pregnancies, you can see a very similar pattern. It's different in that this is a different tissue. It's overall lower methylation than what we saw in cell-free fetal DNA. But again, the obese is hypermethylated compared to the intervention and the controls, which are down below. And we saw that in all 3 of these brain regions, that overall pattern. So this is an interesting region. There's a lot of genes that are not very well characterized. There's some novel non-coding RNAs, but the one that is well known, especially in the human literature, is this miRNA called 663. And in the human literature, this associates with obesity and adipocyte differentiation, immunity and inflammation, the mechanism of resveratrol action, as well as cancer. It's thought to be a biomarker of colon cancer in humans. And the nice thing about this block is that we can then correlate it with a bunch of other measurements from the maternal blood. Remember, we're also measuring all those immune markers and metabolomics. In this case, here we have this positive association over this this block with obesity. We didn't see, there were no significant changes with associations with behavior, although you can see there's some trends. But then if we look out here in the immune markers, the strongest associations were with IL-15 in the first trimester and IL-8 in the second trimester that are positively associated with methylation that's also positively associated with obesity. And then if the metabolomics, again, there were some strong associations for a number of these, including a positive association with acetate and a negative association with formate. The other thing we did, and I think Hung kind of pointed out this too, this systems biology approach to kind of reduce the complexity. We make modules of genes that are co-methylated, and this is commonly done with transcription, and we've developed a new pipeline called CoMethyl that'll do this with methylation data as well. And so basically, I just want to point out this blue module that really came out. So now we're looking at the brain. So this is infant hippocampus from the offspring from the pregnancies. And here you can see all the heat map of the correlations, and you can see there's quite a few. I think it's easier to look at down here. So this is one where the group of genes that are summarized in this eigengene value show lower methylation in the obese compared to the control. And you can see the interventions are in the middle, particularly with calorie restriction brought this level almost up to the level of control. The fascinating thing about this is it also correlated with behavior. So here you can see there's a positive association with the ability of the monkeys to recognize, to remember abstract stimuli, but a negative association with to remember with social stimuli. And this is important for what you see with autism and the distinction of these two memory domains. When you look at the lipids, there was an interesting correlation with linoleic acid, a positive correlation there. And this is Amir Taha is very interested in this group of linoleic acids that are involved in brain development. And then at the level of metabolomics, you can see positive associations with things like asparagine and citrate, and that were also measured in the brain. But then we can also compare those brain measurements with what's in maternal blood. And again, I won't go into all the genes that are in that module, but it's really interesting. This is DUX4 transcription factor. There's a duplication. There's other genes that are regulated by it. And you can see if we just look at the DUX4 cell-free fetal DNA methylation, it was positively associated with this module. And there were also lower levels of cytokines, including MCP1 in the first trimester. And then in the metabolomics, just to kind of summarize, there were some that were grouped into the sort of the one carbon metabolism. You can see creatine here was particularly strongly negatively associated or positively associated with this methylation of this module. And then the tricarbolic acid cycle metabolites also were interesting in terms of showing some associations. And here's, you can see glutamine as a negative association in the first trimester. So for the conclusions from this were that maternal obesity is associated with epigenetic changes to infant brain that correlate with behavior and metabolism. At specific gene loci, DNA methylation patterns in the cell-free fetal DNA derived from placenta correlate with those found in brain. Maternal blood levels of inflammatory markers and metabolites are associated with cell-free fetal DNA methylation levels and detectable as early as the first trimester. And you can see that first trimester actually had some of our strongest associations. So these studies suggest that an integrative analysis of cell-free fetal DNA methylation patterns, perhaps together with immune and metabolic markers, may be useful in early screening tools for neurodevelopmental disorders. And that's where we're going in the future, is taking this back to human cohorts. So I just wanted to, briefly point out that this is the Marbles cohort that we've worked with Rebecca Schmidt, and they have plasma stored, and we're hoping to isolate cell-free fetal DNA and look at interactions there. And then also, this is our new Perinatal Origins Disparity Center, supported by the Office of Research Impact Center, and Leanne Simmons, who's my co-director in that, has this cohort called Grow Well, which is a behavioral digital health pregnancy intervention. And we're going to be trying to do this from blood spots and isolating cell-free fetal DNA from that as part of this new Team Science Award from the UC Davis Health School of Medicine. And I think I mentioned everybody, so I'll stop there and take questions. We're running short on time.
Thank you so much, Janine. So both Janine's and Hang's talks are open for questions. You can either just blurt it out aloud or put it in the chat. There was an intense chat back and forth
Great
going already. Yeah, Erman, please go ahead. Unmute yourself.
Hey, Janine, that was very interesting. I have an interest in cell-free DNA, but more on the quantitative side. So I was curious, what method did you guys use to discern cell-free fetal DNA from maternal cell-free DNA?
Yeah, great question. I didn't have time to show that, but because the pregnancies were all male, actually the reason we got cell-free fetal DNA originally was because they were already screening for male pregnancies in these monkeys, and they were isolating it at the primate center. Originally, we were going to do biopsies, so Cheryl was doing like equivalent of CVS, but they isolated the cell-free fetal DNA from the monkeys for us. But that gave us the advantage. So with the epigenetics, because the X chromosome promoters are completely unmethylated, we could actually use that to figure out, well, for one, with just the genetics, we could look at the X ratio of X to Y DNA in the fraction. And we saw that increased throughout pregnancy like you would for cell-free fetal DNA. We still don't exactly know like an exact percentage of what we were getting, but it was enough to convince ourselves that yes, this is at least, I mean, probably what we're looking at is a combination of the maternal and the fetal, to be honest. But we knew that, yeah, we have a nice graph showing with this estimate of the XY ratio, we see an increase in the what we know is fetal. Probably the elegant way to do this is to know the genetics and to compare the maternal genetics to the fetal genetics, but we didn't have whole genome on the monkeys. But in some of the human studies, we may have whole genome, and that would be more feasible.
Well, one possibility would be using single nucleotide polymorphism. And
right, yeah. Right, or if you had, I guess, array data, SNP arrays from both the mother and child.
Yeah, that's very interesting. I have a question for both of you. So just to consider epigenetics and that you very nicely presented at the beginning of your talks, what epigenetics plays in the interface of nature versus nurture. Are you only studying DNA methylation, or are you also interested in looking into histone modifications that are also considered to be epigenetic regulatory pathways?
Yeah, I think I showed a nice example of this, and she can, yeah, but we always look at, see, it's easier to get DNA than it is to get chromatin. You need a lot more cells to get chromatin. So DNA is wonderful. Like we can get it from newborn blood spots now. We can get it from cell-free fetal DNA. So you just need DNA, but then you can work your way back. So I know Hung showed this, and we've done this with all of our studies too. Once you have these DMRs, then you can look at the chromatin state maps. So you're not directly looking at the histone modifications, but you can say, okay, this region that's differentially methylated is an enhancer in, brain or liver or whatever your tissue. I mean, you compare, because those maps are already there. So we use the existing epigenome roadmaps, which is wonderful for that. So I think it's almost easier to do it that way. We have done some other marks. There's things like ATAC-seq, which gets at the open chromatin, and that can be very useful. And there's a lot of great. We're also moving to single cell levels. We've done some single cell transcriptomes that haven't quite moved to the methylomes, but we have collaborators that are doing that. There's some great new methods for getting at single cell methylomes or single cell ATAC-seq. So that's kind of the future.
Yeah, that's really fascinating. And one more thing that I've been always wondering, that's about, because both of you mentioned the relevance to inflammatory genes and showed some great effects of environmental exposures on inflammatory gene regulation. What about the glucocorticoid receptors? So glucocorticoid receptors, tissue-specific expression, even under homeostatic normal situation, is driven by epigenetic, especially DNA methylation. So have you seen anything related to the
We haven't seen that either in a gene or a pathway yet, in at least the models we've looked at, but I think that is relevant for, I don't know if Han has.
Not in wildfire, but I think we did a single cell RNA-seq analysis following like a house dust mite treatment. We did a single cell analysis of the whole lung or the epithelial cells in mice. We did see the glucocorticoid receptor gene pops out. But in terms of its relation to, all the other exposures, I mean, we haven't had the luxury, as Janine mentioned, haven't had the luxury of doing single cell for the, the larger human studies or even, studies in rhesus monkeys yet. But like, I guess going back to your other questions, I mean, if you collect peripheral blood, then maybe that's a good place to start from to do histone modifications. And also all the other like ATAC-seq, chromatin accessibility. I think that's probably one way to go. But the cells do have to be properly stored, which is not usually the case for large studies or human studies. Like you have to basically collect the PBMCs and store it in the right way instead of freezing down the whole blood.
Thank you so much. It's been really wonderful. We were privileged to be able to listen to your studies. And everyone stay tuned. We have very nice presentations lined up in this series. And please look out for announcements from Ana Lucia. And thank you, everybody. Have a great weekend.
Thank you. Bye.
Bye.